Good morning students. Welcome to Leela's tutorial. So in today's class we will discuss about RFLP. So RFLP is nothing but restriction fragment length polymorphism. So in last class we have discussed about molecular markers. What are molecular markers? These are small fragments of DNA which are taken from the sample in order to identify or differentiate the one organism from a group of organisms. Otherwise, the DNA sample can be differentiated from a pool of DNA, right? So here, what is the technique involved? Let us see. So here, the restriction fragment length polymorphism, the name itself implies that the restriction fragments, that means the restriction enzymes are used in order to fragment the DNA or in order to cut the DNA into different length or sized molecules. Right? So these different sized molecules are used in detecting or differentiating one organism from another organism. So see here, in RFLP, different size or length of the DNA fragments are produced by digesting the DNA with restriction enzymes. What is the use here? The first one is identification and differentiation of organism by analyzing unique pattern of their genome. So I told that restriction sites are present on the genome of each and every organism at different sites. So when you add the restriction enzyme to the genome, they go and cut at that specific restriction site, creating different sized molecules. These different sized DNA fragments will not be same in every individual. Right? It differs from one organism to the another organism. So in that way, you can know or identify that particular organism from the complete gene pool or a sample. Otherwise, you can differentiate that organism from another organisms. Right? In the second use, it is to know or identify the recombination rate in the loci between restriction sites. So here, the recombination, when the recombination occurs, where it occurs in chromosomes. If the recombination or the crossing over occurs in between the sister chromatids, there will not be any change because they have the same pattern, right? Whereas if it is in case of non-sister chromatids, the recombination results in exchange of the chromosomes or the exchange of the genes. If these exchange patterns has occurred, right, you want to know in which chromosome this recombination has occurred or at which loci or which site. In that case also, by using your RFLP technique, you can know the location, right, location between the restriction sites and know in which chromosome the recombination has occurred and at what rate it has occurred. Then the next one is detection of the genetic diseases. So here, if you know the complete genome of an organism, you go for the gene mapping. Gene mapping is nothing but the knowing the distance between the successive genes, their arrangement and also the complete mapping. That means you know at which position the genes are present and what genes are present. So in this alignment, when you again go for the genome analysis in a diseased person, this alignment will differ from the natural alignment, right? In that case, you can know that in that particular gene, right, there is a variation which is responsible for that disease, right? Then this can be treated by using your gene therapy, where you can correct the gene, right? And even in case of sickle cell anemia also, you can use this RFLP and also for mapping of the genome of any organism. Then coming to the technique, the first point, here you are supposed to take the DNA sample. Here I am taking 3 samples from 3 different organisms, right? If I am taking 3 different samples, the DNA in an Eppendorf tube, I am going to add the restriction enzyme. Right? When I add the restriction enzyme, these enzymes act as a molecular scissors and cut the DNA fragments at their specific sites. Right? They cut the DNA at specific sites, resulting in restriction fragments. That means the different sized or length fragments are created. And these restriction fragments of the DNA are then loaded in the gel. Right? So here I am taking agarose gel where I am going to perform the electrophoresis. Right? So here I am going to have 3 wells in which I am going to load sample A DNA, sample B DNA and sample C DNA after restriction digestion. That means I am going to add these restricted fragments in A, B, C respectively. Right? Once I set up this electrophoretic tank and undergo the electrophoretic separation of these fragments, I told the restriction enzymes has cut the DNA into several fragments, right? At each and every restriction site they have cut. So as a result, there is formation of different sized fragments, right? The size of the fragments is differing. The restriction enzyme, so if it is cutting, these are the restriction sites. If I am adding the restriction enzymes, what happens? They go and bind at that particular restriction sites and cut the DNA, right? When they cut the DNA at that, so here we get different sized fragments, right? We get the different sized fragments. So these different sized fragments, when you go for the electrophoretic separation, they get arranged based on their molecular size, right? That is the length of the DNA. The smaller size will move at a faster rate through the pores of the gel and they reach to the next corner, right? So here you have, they are arranged in a pattern in such a way that the larger size molecules are here and the lower size molecules are towards the other end, right? So once there is an electrophoretic separation of the bands, now as you can't store this gel for the longer time or you can't go for the further studies, it is very fragile to handle. So what you do? You have to transfer them onto the nitrocellulose membrane paper, right? So that the paper is easy to handle and it will not react with anything and it can be stored for longer time. So here, see this setup. So here in this, in a tank, I am taking the alkaline solution, then the sponge. Over this sponge, I am going to lay this gel over this sponge, right? Over that, the nitrocellulose membrane is placed. Over the nitrocellulose membrane, I have placed the paper towels or the tissue papers. I am going to put some weight on them. So due to the capillary action, the buffer will rise up through the sponge and the bands which are present in the gel are moved from gel to the nitrocellulose membrane, right? Once the complete arrangement is done, here the nitrocellulose membrane now consists of the bands. The bands are blue in color because of the tracking dye. We have added tracking dye to the DNA sample here, right? So now once you are adding here, you can see the DNA pattern bands have been transferred from the gel to the nitrocellulose membrane paper. Now you have all different sized fragments here. But what is RFLP? You want to know the particular site or the loci, whether that restriction site is present in that or not, or how many number of restriction sites are present in them. So here now you want to know one particular DNA site. So what you do? You create a probe. The complementary DNA which you are going to tag them, right? You are going to create a probe and label that probe with radio labeled element, right? And those radio labeled probes are allowed to be immersed or allowed to be placed inside the plastic cover or a box placing this nitrocellulose membrane. So now you have the nitrocellulose membrane which is completely immersed in an radio labeled probes solution. So after incubation, as the probes are complementary to the specific DNA site, whatever you want, only that complementary DNA molecules you have created. So they go and bind to those complementary DNA molecules. So after the incubation, you will take out this nitrocellulose membrane, wash it in wash it so that untagged probes. So you see, in case these are the radio labeled probes, right? So radio labeled probes are specific acid. So this will go and bind to the specific DNA. If this is not the specific site, they will not go and bind. So what happens when I wash them? The unlabeled probes are unbonded. There is no bond between these. So these will be washed off. On only the sites which are specific, they go and bind. Right? So after washing, here the last step is I am going to take this nitrocellulose membrane paper. See here, a photographic film is laid on the top of the paper and exposed to radioactivity. So when I expose to the radioactivity, the radio labeled probes will show the image. Okay, you can see here. See the different pattern. All the DNA fragments are not showing the image. Right? So only the radio labeled probes which are tagged or bonded to the specific DNA, only those are shown in the image. That means these are the probe in the band forms an image corresponding to the DNA. The specific DNA, whatever we want, only to that specific DNA, these complementary radio labeled probes have gone and tagged to it. Right? Because of which you can see the image over the photographic film. So like this, you can confirm or you can compare. See, if the bands are same, that means what? Both B and C samples are from same animal, right? So here this is the A animal which is not at all matching with the B and C. So like that you can compare and also you can analyze the particular DNA sample from a pool of DNA. Is that clear? So here the molecular biology and the biotechnology uses this RFLP technique very widely, right? So all the techniques require these restriction enzymes, and this RFLP is further modified, which we'll discuss in the next class. Thank you. If you have any doubts, you can ask me, and please subscribe for further videos.